Hi everybody, and welcome to our module on hemoglobin. Let's start by talking about how oxygen is transported in our blood. There's a very tiny amount of oxygen that dissolves in our plasma, and the amount that's dissolved is determined by Henry's law, which you probably studied in a college chemistry class. Henry's law says that the partial pressure of oxygen times the solubility of oxygen in water will equal the amount that's dissolved in liquid like plasma. And this turns out to be a very tiny amount of the total oxygen in the blood. If we didn't have hemoglobin, we would have very little oxygen carrying capacity and we would never have evolved to become the complex organisms that humans are. Luckily, however, we evolved hemoglobin, which carries 98% of the oxygen in our plasma, and this allows us to deliver much more energy to cells of the body. Hemoglobin is made up of globin chains and heme. Globin chains are large globular proteins, and there are several different kinds that are used to make the molecules found in our red cells. There are alpha globin chains, beta chains, gamma chains, and delta chains. And all hemoglobin molecules are made up of 4 chains in 2 pairs. The other component of hemoglobin is heme, which is a molecule. It's not a peptide. This is its structure shown on the screen here. It's made up of nitrogens and carbons with an iron in the center. And this iron is what is going to bind oxygen. And this ring that surrounds the iron is called a porphyrin ring. There are 3 major types of hemoglobin found in the human body. Hemoglobin A is the adult hemoglobin. It's the most common form of hemoglobin found in adults. And it's made up of 2 alpha chains and 2 beta chains. Just like I told you before, all hemoglobin is 4 chains in 2 pairs. The second type is hemoglobin A2. This is also an adult type of hemoglobin, but it's less common than hemoglobin A. This makes up only about 2 to 3% of adult hemoglobin, and it's made up of 2 alpha and 2 delta chains. And then finally, hemoglobin F is the fetal type of hemoglobin. This is what is found in babies in the womb, and it's made up of 2 alphas and 2 gamma chains. Next, let's talk about oxygen-hemoglobin dissociation curves, which are a classic topic in pulmonary physiology. Shown on this slide is an example of an oxygen-hemoglobin dissociation curve. It plots the partial pressure of oxygen in the bloodstream along the x-axis, and along the y-axis is the percentage of hemoglobin binding sites for oxygen that are saturated with a molecule of oxygen. So for example, if every single oxygen binding site was filled with a molecule of oxygen, then we'd be up here at 100 on the y-axis. And if not a single binding site for oxygen were filled with an oxygen molecule, we'd be down here at 0. And remember, the partial pressure of oxygen is basically a measurement of the oxygen concentration in the bloodstream. So just like you would expect, as the oxygen concentration in the bloodstream goes up, more and more binding sites of hemoglobin become saturated. But here's the thing, it doesn't go up in a straight line like this. Instead, it forms this special curve known as an S-shaped curve. And for step 1 of your boards, you need to understand why this curve is S-shaped like this and not a straight line like this. So each molecule of hemoglobin has 4 heme moieties, and each heme moiety has an iron binding site. Therefore, each molecule of hemoglobin can bind 4 oxygens. And it turns out that the heme groups do not simultaneously bind oxygen. In other words, they don't bind 4 molecules of oxygen all at once. Instead, they bind 1, and then they bind a second, and then a third, and then the fourth. They bind them one after another in series. And not only do they bind them in series, but when the first oxygen molecule binds to hemoglobin, this increases the affinity of hemoglobin to bind a second molecule. Then the second molecule increases the affinity for a third, and the third increases the affinity for a fourth. The result is that the affinity of hemoglobin to bind the final oxygen, the 4th oxygen, is 300 times greater than the affinity for the first. And this phenomenon is called positive cooperativity, and this is what makes the curve S-shaped and not a straight line like this. Hemoglobin is an example of an allosteric protein. The word allosteric means other site. So allosteric proteins are proteins where binding of a substrate at one site changes the likelihood of binding at other sites. Most allosteric proteins are multi-subunit proteins like hemoglobin, which is made up of 4 proteins. So hemoglobin is therefore an example of an allosteric structure, and the cooperativity we talked about in the last slide is an example of a positive allosteric effect. It's called positive because binding of one molecule makes another molecule more likely to bind. There are also negative allosteric effects where binding of one molecule makes binding of another less likely to occur. So now let's go back and take a closer look at what's happening in the body as it relates to the oxygen-hemoglobin dissociation curve. In our lungs, the partial pressure of oxygen is about 100. And so if you look at this curve, in our lungs, you can see that the hemoglobin in red cells is going to become 100% saturated. So that's what happens in the lungs. All of the binding sites basically get occupied by molecules of oxygen. In the tissues of our body, the PO2 is much lower. It's about 40. If you take a sample of venous blood, which is blood that has just left the tissues, and measure the partial pressure of oxygen, 40 is a normal result. So what's happening according to this curve in the tissues is that oxygen is only going to be about 75% saturated or so. So what this means is that hemoglobin molecules move through our lungs and become fully saturated, And then when they get to the tissues, they unload oxygen down to about 75% saturated, and that difference in saturation represents the oxygen released to tissues for metabolism. Next, let's talk about the different formations that hemoglobin can assume. The 4 globin chains in every molecule of hemoglobin can become arranged in 2 different formations. One is called the taut form, the other is called the relaxed form. The taut form tends to release oxygen. This is the favored form of hemoglobin in tissues because it allows more release of oxygen for metabolism. The relaxed form tends to hold on to oxygen. This is the favored form in the lungs because it allows more binding of oxygen. So in our body, if you look at all the hemoglobin, you will find some in the taut form and some in the relaxed form. But the relative percentages of the taut and relaxed form can change by certain environmental conditions. And when this happens, it shifts the oxygen-hemoglobin dissociation curve. And this is a very important concept of pulmonary physiology. So the idea here is that the affinity of hemoglobin for oxygen can change. It's not fixed. Hemoglobin molecules are modified by the environment within the red cells. And when these molecules are modified, you can get either more or less of the taut or relaxed form. And when that happens, when you get more or less of the taut or relaxed form, that changes the dissociation curve. It can shift either to the right or the left depending on conditions. So if you look at the graph on the screen here, imagine that the blue is hemoglobin in its baseline state. If conditions are right, the curve can shift to the right, like this red one here, or to the left, like this green one here. And what's causing it to shift to the right or the left is that conditions within red cells are favoring either more of the taut form or more of the relaxed form. So let's start by talking about a rightward shift of the hemoglobin-oxygen dissociation curve. If the red here represented a normal curve, the blue dotted line here would represent what the curve can change to when there is a right shift of the oxygen-hemoglobin dissociation curve. So to understand the implications of a right shift, let's talk about hemoglobin under normal circumstances first. So under normal circumstances, the partial pressure of oxygen in the lungs is about 100, and this causes hemoglobin to be 100% saturated. When the molecules of hemoglobin get to the tissues, the PaO2 is much lower at 40, and this causes hemoglobin to fall to 75% saturated. So what our body is doing under normal circumstances is unloading hemoglobin from 100% saturated to 75% saturated. When the curve shifts to the right, we still have a PaO2 of 100 in the lungs. We are still fully saturating hemoglobin in the lungs. The partial pressure in the tissues is still 40, but because of the right shift, we have now decreased hemoglobin saturation down to 50. So the rightward shift allows hemoglobin to go from 100 to 50 instead of 100 to 75. And that represents a significant unloading of oxygen, and therefore the rightward shift really helps to deliver much more oxygen to tissues. And I've shown this down at the bottom here. Normally we go from 100 to 75%, a change of 25, but if we right shift the curve, we could go from 100 to 50%, which is a delta of 50. That represents significant unloading of oxygen. And what's shown on this curve is the same thing I just showed you in that table. Don't get thrown by the curve. Basically what we've got here is in the lungs, we have a PaO2 of about 100. In the tissues, we have a PaO2 of about 40. As you can see, we're normally 100% saturated in the lungs no matter which curve we're operating on. Whether we are on the normal curve or the right shifted curve, we're still fully saturating hemoglobin in the lungs. However, which curve we're operating on makes a big difference when we get to the tissues. If we're on the normal red curve, we only get down to 75% saturated. That's a delta of 25. If we're operating on the blue curve, we get all the way down to 50, which is a delta of 50, and therefore we've unloaded much more oxygen. So you can just remember, right curve shifts lead to the release of O2 in the tissues. That's the idea behind this. Hemoglobin undergoes this kind of transformation in environmental circumstances where lots of O2 needs to be released to tissues. So what are causes of right shifts? Well, anything that is associated with rising metabolic activity where the tissues need more oxygen. So how can hemoglobin tell when tissues need more oxygen? Well, one way is if the CO2 level is going up. CO2 is produced by metabolism, so if there's lots of CO2 around, that will be sensed by hemoglobin, and it will change its form to have more of that taut form present, which tends to release O2, and that will cause the curve to shift to the right to look like the blue line here. Anytime carbon dioxide level goes up, the pH inside of red cells falls. I talk about this in the videos on respiratory acidosis. So therefore, low pH is a sign of high CO2, which is a sign of rising metabolic activity. And therefore, low pH is another thing that causes hemoglobin to shift to the right. And then finally, if the temperature goes up, that means there must be lots of metabolic activity around. And therefore, that will also cause a shift to the right of the hemoglobin oxygen dissociation curve. And then finally, let me mention a concept in pulmonary physiology known as the P50. The P50 is the point along the x-axis where hemoglobin is 50% saturated. Anytime the curve shifts to the right, that increases the P50. To understand this, let's look at the graph at the bottom right side of the screen. If we're operating on the red curve, the P50 occurs right here at a partial pressure of oxygen of about 25. If we shift to the blue dotted curve, the P50 occurs here, which is at a partial pressure of about 40, which is higher. So therefore, we can say that any rightward shift increases the P50, which is simply a number used to describe where hemoglobin is in terms of oxygen dissociation. And if you want a simple mnemonic to remember all these things with, just remember that the word right starts with R, which stands for release oxygen, and it ends with T, which stands for favors the taut form of hemoglobin. And if you understand rightward shifts of the hemoglobin dissociation curve, it's very easy to understand left shifts. They are the opposite. Shown on the slide is a normal curve represented by red, and the green dotted line is a leftward shift. This decreases the P50, and left shifts occur anytime there's lower metabolic activity. Remember, they're the opposite of right shift. So if the CO2 level falls, or the pH rises, or the temperature falls, all these things will cause oxygen dissociation curves to shift to the left. And when the curve shifts to the left, it's less likely to release oxygen and more likely to hold on to oxygen. You can just remember this, that left curves latch on to oxygen. Now let's talk about a very important molecule that is found inside of red cells that can affect oxygen uptake by hemoglobin. And that molecule is 2,3-bisphosphoglycerate, or 2,3-BPG. This is its structure shown on the screen here. And 2,3-BPG is always found in red cells at some level, and it promotes oxygen release from hemoglobin. It's a negative allosteric effector. This means that when 2,3-BPG binds to hemoglobin, it causes less oxygen to bind. So high levels of 2,3-BPG are associated with decreased oxygen affinity for hemoglobin, and therefore oxygen becomes released and there's more delivery to tissues. 2,3-BPG is created from 1,3-BPG, which is an intermediate in glycolysis. Remember, red cells do not have mitochondria, so they depend on glycolysis for metabolism. And 1,3-BPG is a glycolysis intermediate, and red cells have an enzyme called BPG mutase, which can convert 1,3-BPG into 2,3-BPG. So in other words, red cells are sacrificing a little bit of ATP, in other words, a little bit of glycolysis, in order to make 2,3-BPG, but that's important because it modifies oxygen uptake. So as I said before, there's always some 2,3-BPG in red cells, but the amount can go up when red cells are exposed to chronic hypoxia. And this should make sense to you based on what we just talked about. 2,3-BPG causes hemoglobin to let go of oxygen. So in the setting of chronic hypoxia, you want oxygen to be released, and therefore that's when 2,3-BPG levels go up. So patients with COPD can have high levels of 2,3-BPG. So can patients who live at high altitude. So can patients with chronic anemia. In all these situations, there's a chronic level of hypoxia in tissues, and that leads to red cells making more 2,3-BPG. So to explain the impact on oxygen binding of 2,3-BPG, let's consider the situation of high altitude. So if you're at sea level breathing room air, the PaO2 you're going to find in the alveoli of your lungs is usually about 100. And then as we said earlier, out in the tissues, the PaO2 is about 40, and that causes the oxygen saturation of hemoglobin to go down to 75%. If you're up at high altitude at Machu Picchu in Peru, the PaO2 you're going to find in your alveoli is much lower. It's about 75. This means that we've moved down the oxygen hemoglobin dissociation curve, so we may not fully saturate hemoglobin. And then when we get to tissues, we're only unloading down to 75%. So that's some oxygen, but not as much as we could potentially release if we modified hemoglobin. And so that's what 2,3-BPG is going to do. It's going to shift the curve to the right, just like we talked about earlier. Now, if we're living at Machu Picchu chronically with hypoxia all the time in our lungs, instead of going from some level here a little bit below 100 down to 75, we're going to go from whatever level we have in our alveoli all the way down to about 50. That's a significant increase in the unloading of oxygen, and that's going to be beneficial if we're living at high altitude. It's also helpful in patients with COPD and patients with chronic anemia, any situation where the body is chronically subjected to hypoxia. So if we go back to our causes of right curve shift, those were a high level of CO2, a low pH, and a high temp. And we can add to this list an increased level of 2,3-BPG. Anytime that happens, it's going to shift the curve to the right. And anytime the level of BPG goes down, it's going to shift the curve to the left. Now let's talk about fetal hemoglobin, which is hemoglobin F. And as I told you before, hemoglobin F is made up of 2 alpha and 2 gamma chains. After about 8 weeks of development, hemoglobin F forms in utero and is the predominant hemoglobin type. It makes up to 90% of the baby's hemoglobin. After the baby's born, the level starts to fall. There's still a lot of hemoglobin F at birth, but it starts to fall steadily, and then in adults it makes up less than 1% of total hemoglobin. The reason this is the preferred form of hemoglobin for babies growing in the womb is because it has a higher oxygen affinity than hemoglobin A. Remember that hemoglobin in the mother is going to be mostly bound to hemoglobin A, so you want a hemoglobin in the baby with a higher affinity so it will pull oxygen from hemoglobin A and bind it to hemoglobin in the baby. And this is very important because the fetal oxygen partial pressure is only about 40. In a normal adult, the oxygen partial pressure is about 90 or so. It's much lower in the baby when it's growing, so it needs a form of hemoglobin that very easily binds to oxygen. So what this means is that fetal hemoglobin is left shifted compared to regular adult hemoglobin. It has a higher oxygen affinity, and if we look at these two curves, the red could represent adult hemoglobin, the green would represent fetal hemoglobin. It's shifted to the left, it has a lower P50, and therefore it binds oxygen with higher affinity compared to adult hemoglobin. And the reason that fetal hemoglobin is left shifted compared to adult hemoglobin is because fetal hemoglobin has altered binding of 2,3-BPG. It's super high yield to know this for step 1. This is what they're going to want you to understand. In adult hemoglobin, 2,3-BPG binds to the beta-globin chains very well. But in fetal hemoglobin, there are no beta-globin chains. There are gamma chains, and 2,3-BPG does not bind gamma chains very well. For this reason, in fetal hemoglobin, there's less 2,3-BPG binding, and that causes the oxygen affinity to go up. In other words, that causes a left shift. Let me briefly mention myoglobin, which shares some similarities to hemoglobin. Myoglobin is a protein found in skeletal muscle and the heart. It's a single chain, not 4 chains like hemoglobin, and because it's a single chain, it can only bind 1 molecule of oxygen. You can create an oxygen saturation curve for myoglobin, and if you do, it will look like this green line here. And you can see that it's very different from the S shape of hemoglobin. And this green curve shows you why we are better off having hemoglobin in our red cells than myoglobin. At the partial pressures found in the lung, like 100, myoglobin is fully saturated. When you move down into the tissues at partial pressures like 40, myoglobin is still nearly fully saturated. So the problem we would have if we used myoglobin instead of hemoglobin is that our cells would never offload much oxygen, and that wouldn't be very effective for metabolism. But because we have hemoglobin, and because hemoglobin has this S-shaped curve, when we get down into the tissues, we unload lots of oxygen, and that's good for metabolism. I'll finish this video by talking about 2 special pathologic conditions that relate to hemoglobin and oxygen binding. The first one is carbon monoxide poisoning. Carbon monoxide is a gas, and it binds to iron in heme with 240 times the affinity of oxygen. When it does, it forms a structure called carboxyhemoglobin. Because it binds so avidly to hemoglobin, it blocks binding sites for oxygen, and therefore less oxygen can be absorbed by hemoglobin. This creates what's called a functional anemia. Basically, even though there is enough hemoglobin, the body behaves as if it's anemic because lots of the oxygen binding sites in hemoglobin are blocked. Not only can hemoglobin bind less oxygen, but it has trouble offloading oxygen. This is because carbon monoxide is an allosteric modifier of hemoglobin, and it shifts the curve to the left, and therefore less oxygen will be unloaded at any given partial pressure. So this slide shows the effect of carbon monoxide on the hemoglobin-oxygen dissociation curve. A normal curve is in blue, and because of carbon monoxide, there's less carrying capacity of oxygen, so the whole curve moves downward like this. In addition, there's a leftward shift, so when you get down into lower partial pressures, more oxygen remains bound than if you were operating on the blue curve. People who are exposed to carbon monoxide poisoning usually have nonspecific symptoms. Headache is the most common, but they can also say that they have malaise or nausea or dizziness. A classic but very rare finding is to have cherry red lips. This occurs because carboxyhemoglobin is red, and so these patients will develop red lips, very different from the blue lips you see in patients who are cyanotic. Now, patients with carbon monoxide have a decreased oxygen content in their blood. However, the pulse oximeter is often normal. That's because it can't differentiate between carboxyhemoglobin and oxyhemoglobin. So it will say that 90% of hemoglobin molecules are saturated. It can't tell you that only half of those molecules contain oxygen, the other half contains carbon monoxide. The way you make the diagnosis is by measuring a carboxyhemoglobin level. A normal level is less than 3%. Smokers always have slightly elevated levels, like 10 to 15%. It's super high yield to know this for your boards. A level greater than 15% suggests carbon monoxide poisoning. And the treatment is oxygen. If you put lots of oxygen into the lungs and into the blood, it will push all those carbon monoxide molecules off, and they will be exhaled, and things will go back to normal. The second pathological condition I'll talk about is called methemoglobinemia. So most iron in hemoglobin is normally in the reduced state, which is Fe2+. But a very small amount is found in the Fe3+ state. This is called methemoglobin. And this hemoglobin is bad because it can't bind oxygen, but usually it's just a tiny bit of our hemoglobin, so it's not a big deal. If, however, a pathologic condition causes excess methemoglobin, this can lead to hypoxia because all the Fe3+ won't be able to bind oxygen. So what could cause an increase in the amount of Fe3+ iron that's found in hemoglobin in our red cells? Well, it turns out there are a number of drugs that can cause this. Local anesthetics like benzocaine are famous for doing this. Most patients who receive these drugs do not develop methemoglobinemia, but a small proportion do. Nitric oxide, which is an inhaled gas, sometimes administered for pulmonary hypertension, can cause methemoglobinemia. And then finally, dapsone, which is a rarely used antibiotic, is also known to cause this problem. The treatment is with a substance called methylene blue. It's a reducing agent, and it converts iron from the 3+ state back to the normal 2+ state, which can bind oxygen. So a typical clinical scenario for methemoglobinemia that you should be able to recognize for step 1 could be an endoscopy patient who receives benzocaine spray to the throat to cause numbness of the throat so that the probe can be passed into the esophagus. After the procedure, the patient complains of shortness of breath. When a blood gas is done, a classic finding is the presence of chocolate brown blood, and this occurs because methemoglobin turns blood brown. Measuring the oxygen saturation by pulse oximetry is unreliable. The results are variable depending on the severity of methemoglobinemia. You can get results that are in the 80s, which represent hypoxia, but you can sometimes also get normal results in the 90s. Importantly, if you give the patient oxygen, that's not going to make them feel better, and that's because the problem is not insufficient oxygen. The problem is that iron is in that Fe3+ state, which doesn't like to bind oxygen. When you measure the PaO2 by blood gas, it's going to be normal because there's no problem getting oxygen into the blood. The problem is getting hemoglobin to bind the oxygen. And you can make the diagnosis by finding an increased methemoglobin level, which can be measured in the laboratory. Besides a patient that receives numbing medicine to the throat, another classic scenario is a premature baby given nitric oxide for pulmonary vasodilation. And that concludes our video on hemoglobin.